Moving into the pancreas, we've already discussed the liver in a previous video, the gallbladder in a previous video. Now it's time to look at the pancreas and its functionality in the digestive system. Now up here I have endocrine function. We've already covered this. We've already discussed this back in the endocrine unit. But just keep in mind that the pancreas works in both the endocrine and digestive functionalities. Now, yes, the endocrine functionality was the pancreatic islet secreting insulin and glucagon. And yes, that does technically relate to sugar regulation in the blood, which is digestion-based. But we're going to leave that back in the endocrine system. But just keep in mind that still is there as one of the functionalities. What we want to focus on is going to be the exocrine function, which is the digestion function. So that means we're looking at the acini or the acinar cells. Now, these cells are going to secrete pancreatic juice. They're going to secrete the actual fluid that contains all these enzymes, that contains these buffering agents. And inside these cells, are these zymogen granules. The zymogen granules will contain the actual digestive enzymes. So that means these acini cells contain these zymogen granules, which then contain the digestive enzymes. So it pretty much looks like this. Each one of these is a singular acinar cell. You'll notice that in the tip here, those black dots, those are the zymogen granules. That's where you have the actual digestive enzymes being present. So all those need to head into this space here to be sent down the duct, heading from the pancreas towards the duodenum. So zymogen granules contain the actual digestive enzymes that we'll be talking about in a second. So what is in this pancreatic juice, this mixture of solution that leaves the actual pancreas? Well, it's a pretty watery solution. Now, but the key thing is, it's watery and it's alkaline. The pH is give or take about a pH of 8. So it's slightly basic. But that's necessary because that basic nature helps to neutralize the acidity of the chyme that came from the stomach. Remember, that chyme from the stomach could be a pH of 2, 3, maybe 4, but very acidic. So you're trying to neutralize that to bring it back up to roughly a pH of 7, or somewhere in that ballpark. Now, there are electrolytes in this pancreatic juice, but most of those are HCO3-. Now, what do you remember about HCO3- bicarbonate? It's a buffer. So it's meant to buffer the acidic nature of the chyme, that acidic chyme. Enzymes, we're going to have amylase, lipase, nucleases. Well, amylases, breaking down carbohydrates, lipases, breaking down lipids, nucleases, breaking down nucleic acids. 3 of the 4 main macromolecules. Now, all 3 of those are produced in active form. You're saying, well, how come they don't break down the cells that make them? Because they need ions or bile or something really there to help make sure they're optimally working. Without bile, without certain ions present, they're not really that functional. But the 4th macromolecule, proteins, the proteases, are secreted in an inactive form. That's because once the proteases are active, they simply digest all the proteins present, which means the cells that produce them, they're made of protein, they'd be destroyed. You wouldn't be able to make further proteases to aid in digestion in the future. So it's important to make sure that you're producing the proteases in an inactive form to make sure then that they get to where they need to go before they're activated. So you're not destroying the machinery that makes them. Because if you destroy the machinery that makes the proteases, you can't digest any proteins in the future. So here's what we're looking at. We have the pancreas up here. Then we have more of a zoomed in view of the duodenum here. Notice, coming out of this major duodenal papilla are going to be 3 main inactive proteases. We have trypsinogen. Now, trypsinogen is inactive. The active form will be trypsin. We have chymotrypsinogen. Chymotrypsinogen is inactive and makes chymotrypsin. And the last one is procarboxypeptidase. Again, inactive. Once activated, it becomes carboxypeptidase. So how do you activate these? Well, notice up here on the brush border, you're going to have membrane-bound enteropeptidase. Those are coming from the simple columnar cells. That membrane-bound enteropeptidase can activate the initial trypsinogen, which means by activating that, it makes more trypsin. Trypsin is the active protease now. Once you have trypsin, it can self-activate the trypsinogen. But trypsin can also activate, if you notice right here and right here, it can activate chymotrypsinogen to chymotrypsin, and it can activate procarboxypeptidase to the actual active form of carboxypeptidase. So you have to initially use the membrane-bound enteropeptidase. That activates the inactive form of your trypsinogen. Then you make trypsin. Then trypsin is self-activating and can activate the other two enzymes as well. So it's being secreted from the pancreas. Yes, it's coming down that ductwork, but it's all inactive. It's activated inside of the small intestines by the simple columnar cells producing the membrane-bound enteropeptidase. So how do you regulate this? How do you control this? Coming back to CCK again. And we're going to bring back secretin. Now, CCK, if you notice, is the pancreatic juice being produced by the acini. This is the enzyme rich. So CCK is really helping with the enzyme production. Well, secretin, if you notice, is bicarbonate-rich juice here. So they're both important, but they have two different stimulations here, CCK and secretin. Now there's a lot here, so I'm going to blow it up full screen. First off, what you'll notice is the initial chyme right here being secreted from the stomach into the small intestines. That then is going to cause a release of CCK and secretin. Now next up, the CCK and secretin are being released, and they're heading into the bloodstream, which means they can go to every other part of the GI system. CCK, as we mentioned, is going to induce in the pancreas here the enzyme-rich solution. While we know secretin, which is also here, secretin's going up and inducing the bicarbonate-rich. But they can also then, up top here, stimulate in the liver. What happens now at this point is we're having secretin transported there to the liver, helping produce bile more rapidly. You then also come down to the gallbladder, remember? We talked about CCK there, causes contraction of the gallbladder. And finally, during your cephalic, your gastric phases, the vagal nerve stimulation is going to be helping out. So you're having vagal nerve stimulation to really help move this whole process along. So really what you're seeing here is CCK and secretin aren't just acting in the actual small intestines or just the pancreas. It's also heading up to the liver and the gallbladder. Those two chemicals are helping with the entire system and its functionality. So a little bit about the pancreas, some of the enzymes it produces, and how some of those enzymes are activated.